Let's go ahead and work on this beauty. We're going to investigate the central dogma of biology from various perspectives. We'll start off with a standard flow chart where we start off with DNA at the top, DNA in the form of a gene. DNA is transcribed into an mRNA transcript. You'll notice I'm using blue for DNA, purple for RNA, and then I'm going to use green for the next level, which is amino acids and proteins, and that will hold true throughout this video. An mRNA transcript is then translated into a sequence of amino acids which are then folded into a protein. They will always fold the same way given that conditions of temperature and pH are within homeostatic range. A helpful way to study the central dogma of biology is to look at a cellular model and investigate exactly what's happening within the cell. Now, we have not studied the cell in a great deal of detail, nor are we going to, but there are a few parts of the cell that you really should know. So we're going to start off, you'll notice that of course that I use that bright orange color to denote the cell membrane, the plasma membrane, indicating that these are those phospholipids, the fatty acids, which we drew in another video in a bright orange to really differentiate them and pay attention. All right, so you'll notice I drew a strip of DNA in blue. And I'm going to start to draw the nucleus around here. The nucleus has a few roles, but the main role of the nucleus of the cell is to house the DNA and to protect it. There is actually a double layer of phospholipids there. So I drew it in orange, so it's phospholipids, but it's a bilipid membrane doubled. So there's actually two layers. Very protective. Okay, so there's our nucleus, and now I'm drawing the arrow to indicate that we're now undergoing transcription. And there we go, there's our mRNA transcripts. You can actually have multiple mRNA transcripts coming from one gene. I'm kind of showing the gene there in brackets, in black brackets. There are two basic ideas that I want you to keep in mind with transcription. We're not looking at all of the enzymes that are involved in the unzipping and the 3' to 5' and the 5' to 3' and the RNA polymerase and all those things. But I do want you to kind of pull yourself back from all of those details and think about the idea that in order for a gene to be transcribed, you have to have proteins called transcription factors. When transcription factors are present, you begin to transcribe one particular gene. It basically runs along and zips along the gene and allows for these. And you notice that I drew 3 of them. The reason I drew 3 is that you can continue to produce mRNA transcripts as long as the transcription factor is present. So the longer the transcription factor sticks around, the more mRNA transcripts you will have. This is incredibly important. This actually kind of allows us to develop different characteristics within our cells. This idea of the transcription factors, the concept of transcription is going to be one that we're going to be coming back to. Next up, the mRNA transcripts will leave out of the nucleus and they will be present either in the cytoplasm or attached to the rough endoplasmic reticulum, the RER. The rough endoplasmic reticulum is rough because it has tons of ribosomes along its surface. So whether this next step, which is translation, whether translation occurs in the cytoplasm or attached to the RER, just depends on the circumstances of the translation of that particular protein. I have chosen to simply have translation present in the cytoplasm. Now in brown, I attached several ribosomes along the length of this mRNA transcript. And the purple arrows indicate the directionality in which this mRNA transcript is being translated. It's being translated again and again and again with a leading edge to the right and the lagging edge to the left. So if you think about it temporally in time, the leading edge is going to be the very newest translation occurring, and the lagging edge is going to be the oldest. So your longest translation material is going to be coming out at that lagging end. Hopefully this makes sense. So one mRNA transcript can actually produce about 200 proteins. And I went ahead and packaged one into a Golgi apparatus, a Golgi complex, which also is membrane bound. It's membrane bound with a phospholipid membrane, so it is orange. The Golgi complex is responsible for adding glycoproteins or packaging or the final folding of the protein, and then packaging it and sending it where it needs to go. So this is an organelle of the cell. And what I'm going to do is I'm going to show two hypothetical situations, one in which a protein might be embedded in the cell membrane, and one in which it might be extruded from the cell via exocytosis. So an example of a protein which might be embedded in the membrane, of course, would be, for example, the transmembrane proteins, which we observed in another video. So this one is a receptor. An example of a protein which might be removed from the cell and produced by the cell would be an enzyme, such as lactase. Although we're not going into a lot of details about exactly at a molecular level how transcription happens, we are going to be focusing closer on how translation happens, because what we're really interested in is that end product and producing the protein. So I think it's a little more interesting to focus in on producing the end product at the end of the day. Although transcription is really, it's the more important part of this. Translation is what gets us where we need to be in our class. All right, so we're going to be, again, looking at that leading and lagging end, the leading end off to the right and the lagging end to the left. And I'm only focusing on a smaller portion of this mRNA transcript because otherwise the folding of the protein gets to be a little more complicated. So what I wanted to do, I wanted to show you this a little closer up to make it clear that on the mRNA transcript you have the attachment of ribosomes. And then what happens is basically the ribosomes begin to produce a line of amino acids. They're basically excreting these amino acids, and we're going to try to follow the shape that I drew originally as faithfully as possible. This green color looks super bright until it dries, and then it gets a little bit more reasonable to look at. Okay, so you'll see then that there are 4 ribosomes in a line. As you follow the lagging end, the amino acid sequences get to be longer and longer, and you can actually see it begin to fold on itself. Start to see that secondary folding, if you will, perhaps an alpha helix being started here. Well, next we have to think about, in a practical sense, if you were given a gene and you understood the sequence of the gene, then how is it that you'd actually be able to interpret that gene, transcribe it, translate it, and have some understanding of what the eventual protein would be? So that's what we're going to be doing next. And up in the corner here, I'm going to do a key kind of squinched up in there. I'm going to have DNA, I'm going to have our mRNA in purple, there, DNA in blue, mRNA in purple, and I'm going to add a pink one, tRNA, which we will be coming across, and I'll keep adding to this key. Let us draw a double helix of DNA. We're going to pull out one gene here. We're going to take a look at it. Of course, we're simplifying the story a little bit. Oftentimes, genes will have exons and introns and splicing and so on, but we're going to pretend like it's super straightforward and simple for the sake of being able to teach the lesson. My suggestion is you go ahead and write the letters exactly as I've written them here for a particular reason. You can certainly give yourself a little test and use hypothetical letters or look up some sequences online if you want to practice later. But for the sake of our lesson today, go ahead and just write the letters exactly as they are here. So when we're looking at a letter here, A, T, G, C, what we're talking about, that is the nitrogenous base that is attached to each nucleotide. A nucleotide is a monomer that makes up this very, very long polymer of DNA. A single nucleotide will have 3 parts. Have a pentose sugar, either deoxyribose or ribose, depending if you're looking at DNA or RNA. And then it will have a phosphate group. And then it will have, the only variable part of a nucleotide is actually this A, T, G, or C. Adenine, thymine, guanine, and cytosine. The gene could be much, much longer than this. I use that double line to indicate that we've skipped a great deal in the middle. We're only showing the beginning and the end of the gene. Right, so I've written the word triplet there. We're going to be looking for triplets, which means we're looking for 3 letters together, and we're going to be starting with these 3 here. TAC is always going to be at the start of, that I know of, of every single gene. You're always going to have TAC actually beginning the thing off. So you're going to be following the letters down until you find a TAC, and you know that that's the beginning of the actual transcription that is going to be occurring. And now it is time for us to begin our key up at the top here. We're going to write the letters TAGC, and then we're going to show the complementary RNA that will be developed through the process of transcription. So T goes with A goes with U, G goes with C, and C goes with G. So thymine attaches to adenine, adenine attaches to a new nitrogenous base called uracil. Guanine attaches to cytosine, and cytosine attaches to guanine. So let's begin to create our mRNA transcript. We skip all of the letters leading up to the TAC. That is the first one that we're actually interested in. So we're going to write the pairing down beneath in purple, indicating that this is our mRNA transcript. TAC becomes AUG. CGC becomes GCG. Go ahead and pause the video at this point and see if you can finish out the transcription of these letters. As simple as this exercise is, in my opinion, it's quite easy to make a mistake. You'll see in a little bit I'm actually going to be making a mistake when we're doing the translation exercise. So take your time, make sure that you're really creating the proper, faithful transcription, the mRNA transcript. Again, we have those double lines in the center indicating that we've pulled a lot of the bulk of this mRNA transcript out so that we're only looking at the beginning and at the end. The next step is to look at our codons. So codons, again, we're looking for those 3 letters paired together, starting with AUG. Every time, it always starts with AUG, and then just working our way down the line and separating out 3 letters at a time. The reason we look at codons is because the codons, it's worth emphasizing this point because we're going to be looking at lots of 3-letter things. The codons on the mRNA transcript is what we use to look up the proper amino acid in the universal genetic code. You can find the universal genetic code in your textbook or simply Google it online. It's always the same. So what happens is these 3 letters together, the codons that you find on the mRNA transcript, will correspond with an amino acid, and it always is exactly the same way. That's basically what we're doing here is translation. I'm going to show you a little bit more details about translation. But yeah, you go from the codon at the mRNA transcript, and you'll be able to find the corresponding amino acid. So we can translate this mRNA transcript at this point using the universal genetic code. And again, I've chosen not to indulge in a lot of details in how exactly transcription occurs, and we're not looking into a super detailed level of how translation occurs either, but I thought that looking a little bit closer at how the mechanics of translation works might be helpful in having that overall overview perspective. Some details are really helpful sometimes. So what we're going to start off is drawing just those first 2 codons, the AUG and the GCG. Remember where we found these from? They were originally on the mRNA transcript. And now what we're going to do is we're going to go ahead and draw the anti-codon. We're going to pull out a pink marker and we're going to show how a tRNA is going to pair with mRNA. We're going to find another complement here. A is going to pair with U. U is going to pair with A. C is going to pair with G, and G with C. So one of the ways that DNA differs from RNA is that RNA tends to be, there's a few differences there, but one of them is that instead of thymine, we have a different nitrogenous base called uracil. So now we just complete the pairing here. A becomes U becomes A, G becomes C, G becomes C, C, G, G to C. And next we're going to go ahead and draw this kind of unusually shaped structure called tRNA, which stands for transfer RNA. Where this anticodon is found is basically on one end of the tRNA. And stuck onto the other end of the tRNA, the transfer RNA structure, is an actual physical amino acid. I tell you, I'm having a hard time finding the right green marker. Apologies, because these little amino acids here are being written quite light. However, we're following the same sequence of amino acids up above, so it's MET and then ALA. And so, sorry, but we're basically follow the mRNA transcript that we already created, and you'll be able to see which amino acids we're working with. Remember, when we're looking at the universal genetic code, we're looking at the original mRNA transcript rather than the anticodon. We use the codon to determine the amino acid sequence and not the anticodon. I went ahead and drew a ribosome. It's not quite as simple as to say this happens within a ribosome. It's actually two parts of the ribosome that come together with the tRNA and with the mRNA transcript, and kind of continues to create this structure. But I thought I would just go ahead and draw a ribosome around it to indicate that yes, physically this is happening within the cell. It happens oftentimes within the cytoplasm or in the rough endoplasmic reticulum. But either way, there's always ribosomes involved in translation. Now what we're going to do is blow it up a little bit more. We're going to blow up our amino acid sequence. We're only going to be showing the first few and the last few, with a couple of double lines there to indicate that we're cutting out a bunch of information in the middle. Amino acid sequences can be between 50 and 2,000 amino acids long, so it's only helpful to work with a small number of them at a time so that we can teach the lesson. The first three were methionine, alanine, and isoleucine, MET, ALA, and ILE. You notice that the immediate folding of the alpha helix begins, that secondary folding begins immediately and naturally. The last three were, and this is just looking at our original sequence up above, glycine, proline, and threonine. GLY, PRO, and THR. The last one is going to be a stop. So you see that the original codon is UGA. There's three different codons will indicate a stop, and UGA is one of them. It's actually not encoding an actual amino acid. It just indicates that this is the place where we are no longer going to be transcribing. The end of the relevant information has come. We are now stopping transcription or translation, however you want to look at that. So to give you a sense of the mechanics behind this, the details of exactly how this actually works, I'm going to go ahead and leave the tRNA attached to those very last two amino acids, proline and threonine. Remember, that's where they came from in the first place. They were attached to these tRNAs. We do have the ACU. Oh my gosh, I wrote ACT. This is so embarrassing. This is the kind of mistake that you can make when you're distracted and not properly paying attention. Use my mistake as a lesson. You would not have an ACU, you would not have an ACT, you would have an ACU. I have made that correction, thankfully. Okay, I didn't leave my mistake. That's the important thing, right? Making mistakes, but then correcting them, yes? All right, so our very last three codons were UGA, which indicated an anticodon of ACU. That was our stop tRNA, which again did not indicate an actual amino acid. Our next codon back from that, if you will, was ACU, which indicates an anticodon of UGA, which also indicated an amino acid of threonine. And then the one before that was the codon was CCA. The anticodon is GGU. The amino acid is proline. And so I'm showing that only two of them are being worked with at a time, and then the previous Anticodon was CCC, which indicated glycine. However, that process has already been done, and so the tRNA is now leaving without its amino acid. So what happens inside of a ribosome is you basically work with about 2 to 3 at a time, and they start to shift over as the new one comes in, the next one leaves. And so the two that are being worked with most intensively, what occurs is the peptide bond is formed between them. And then once that is formed, the tRNA releases. Our final lesson today is going to be a discussion on epigenetics. Epigenetics are discussed in different ways depending on the context or the specialty being observed at that time. But in our context, I think the best way to describe epigenetics is basically gene expression. We can either upregulate or downregulate a gene, which at the end of the day, hopefully what you're taking away from this is that at the end of the day, a gene creates proteins. And the proteins then are responsible for physiological functions inside of our cells. So when we upregulate a gene, we create more proteins. And when we downregulate a gene, we create fewer. It really is as simple as that. It's a simple idea with massive implications for the way that cells behave and even in the way that organisms change over evolutionary time, because some of these epigenetic changes can be passed on to offspring. We now know that you don't have to wait for some random mutation to change your behavior, although that does sometimes happen. It's pretty rare, honestly. So the gene doesn't have to be mutated in order to change the cell's behavior. It can simply be expressed more or expressed less, and this leads to major changes. It may be helpful to simply state this as a fact and put it out there. Every cell of your body, no matter how different they look from each other or how different they behave from each other, every single one of them has the same DNA and the same genes, the same genome found in all of them. The difference is the gene expression. Only some genes will be transcribed in an epithelial cell versus a nervous cell versus a muscle cell. Only certain genes will be needed by those cells in order to do what they need to do. So only some genes will be transcribed in each cell. An epithelial cell that is close to the surface of the body will start to produce a protein called keratin. It will enable the gene for keratin to transcribe and translate keratin. Whereas, most of the cells of the body actually don't produce keratin, even though they actually have the gene for it. Now, if you look at our depiction here at that cellular model in the center, take a look at how I showed, two different protein outputs here, one of them being an enzyme or one of them being a receptor. Let's talk about the enzyme and it's important to say these were two different hypothetical situations. Even though I'm showing arrows coming from the same protein, you understand these are two different proteins. Let us begin with the protein that was actually exocytosed from the cell, the enzyme. The one that I want to focus in here on is lactase because it's pretty well known. Lactase is produced only by a small subset of cells in your body that are found in the small intestines. And what lactase does is it allows you to break down a milk sugar called lactose. Now lactose, it's actually not natural for us into our adult years to continue to drink milk. So the default is actually lactose intolerance. Many countries of the world, many cultures of the world, they don't really consume dairy products, and so they do not have the ability to produce lactase. We, Wisconsinites, for example, who cannot live without cheese, we come from a long line of people who also loved cheese, and it's only a few thousand years that certain subsets of humans have continued to produce lactase into adulthood. We have upregulated the gene for lactase in order that we can still eat ice cream and cheese. So this is actually a really good example of evolution, the ability to pass on, the ability to continue to upregulate lactase from generation to generation so that we can have this kind of unique trait. Babies are supposed to eat dairy products. Adults are not. I mean, it's not as much to say we're not supposed to. We're basically saying this is a widely available food product. We'd like to have access to it. Therefore, we're going to evolve. Yes, we're going to evolve the ability to actually consume it without having to involve bacteria that might give us a little bit of pain in our guts. Now let's turn our attention to the receptor that is shown embedded in the membrane. This, of course, is a transmembrane protein called a receptor, and as we've stipulated earlier, a receptor is a protein that receives something. I've thought a lot about which type of receptor I wanted to discuss, and I think I've settled on the oxytocin receptor because it is a story that's going to recur several times throughout the course of this class. So the oxytocin receptor is sensitive to oxytocin, and a cell that produces oxytocin receptors will respond to the presence of oxytocin. And there's quite a lot to say about this, but one of the easy ways to describe how this works is the oxytocin receptors are produced by several different types of cells in your body. Oxytocin receptors, for example, are produced in the mammary glands and help with milk production. Oxytocin receptors are also found in the myometrium of the uterus, which allows for us to, this actually is what allows for the birth process to continue, basically for labor pains, the squeezing of the muscle of the uterus to help expel the pregnancy at the end of pregnancy. Oxytocin receptors are also expressed, remember we're looking at gene expression, they're expressed in various parts of the brain, and they help us to bond with other humans, usually, other organisms. So for example, when a baby is born, generally speaking, a mother will produce tons of oxytocin, and then the oxytocin receptors in various parts, the various cells of the body that are expressing oxytocin receptors, they will all respond to that, and that helps with bonding between mother and baby. There is a push and pull oftentimes between the production of a particular hormone and the expression of the receptors that receive that hormone. So oxytocin is a good example of that. When a person, it's oftentimes called the love hormone, and most endocrinologists will really frown at that description, but I like to use it in my class because it is kind of how it works, and it's pretty easy to relate to this. When you are in love, when you first meet somebody and you start to have those feelings of love, generally speaking, it's a number of different feel-good neurotransmitters, one of the primary ones being oxytocin. So a person who has lots of oxytocin receptors and they're releasing lots of oxytocin will feel really good in the presence of that other person. So the presence of the other person allows us to have the oxytocin release, and especially, I would say probably physical touch helps with that as well. One might think that this is kind of an evolutionary carryover between in the mother-baby bonding situation. So to recap, lactase produced in a very small number of cells in the small intestines, oxytocin receptors produced in a wide variety of cells in different tissues throughout the body, and actually kind of having different meaning depending on which tissue is expressing the oxytocin receptors. It's worth reminding you that every cell of your body has the gene for lactase. Every cell of your body has the gene for oxytocin receptors, and it depends on how much those genes are expressed, how much they are transcribed, and how much they go through the transcription and translation process, and then are either released from the cell or embedded in the cell membrane or whatever has to happen. Only those cells that actually express that and go through that process will have those proteins, and other cells will not, even though they have the genes for them.